Welcome to the GEOTOP-A seminar. Our speaker today is Agnese Barbensi, who will tell us about topologically steered simulations and the role of geometric constraints in protein knotting. Take it away, Agnese.
Thank you so much. Thanks for having me here, and thank everybody for coming. And sorry about the unusual time. I know it's very early for some of you. All right, so let me start with a few disclaimers and intro things. First of all, I want to say that this is a joint work with A.Klotz and D.Goundaroulis. And also, so this is still unwritten work, is pretty much on progress. We're wrapping up the results, but it's still, kind of fresh. That's why this presentation is going to be a little bit more chronological, if you want, rather than result-oriented. I opted for a storytelling, which is more, chronological. So maybe it will sound a little bit, unpolished, but I'm hoping to take you along with me from the beginning and showing you things as they happen. All right, let me start, and please interrupt me anytime with questions. I want to start by this works. All right, I want to start by giving a very quick introduction and motivation to this, if you want, biological problem. So I'm assuming most of us know what proteins are, but regardless, proteins are these huge, big macromolecules. They're often considered the building blocks of life. And they're these big, complex molecules folded to form complex 3D shapes. And one of the perhaps simplest or easiest way to think about a protein is by forgetting about all the complex biochemistry, biophysics of the system, and just focus on the chain of amino acids. So proteins are built by amino acids stuck upon each other, forming a chain and then folded to form these complex 3D shapes. And if we forget about all the 3D features of these amino acids, when you only focus on their spatial positions, we can think about a protein as a curve in space, as a spatial curve, as suggested here by the figure. Now, and that's the point of view we're going to take today. So we are going to think about a protein as a piecewise linear curve in space spanned by the backbone of the protein. So for anyone that has been working with complex, long curves in space, it shouldn't come as a surprise that often, these long, complicated curves are knotted. They often create some form of entanglement. So a natural question at this point is, what about proteins? I'm just telling you that I want to think about them as long, complicated spatial curves. So are proteins knotted? The first protein structure featuring a knot was found by Richardson in 1977, but it wasn't until 1996 when Mansfield performed the first in-depth investigation of knots in protein structures, finding several of them. However, all of the proteins found at that point were shallow, meaning that they were loosely formed, and small perturbations might result in unknotting, as suggested here by the figure. So by shallow knotted, again, I mean the knot is there, but it's loosely formed. It takes most of the chain, and again, it's somehow not very robust. This situation kind of changed significantly in 2000 when Taylor found the first deeply knotted protein structure. So a protein forming a knot that kind of look like this figure here on the bottom of the slide. So the knot is well inside the protein chain and is very robust. It takes only a portion of the chain, and the two ends are very far away from the entanglement. So even small perturbations wouldn't change the fact that the structure is knotted. All right. Then we now know that roughly 1% of the approximately 200,000 experimentally determined protein structures are knotted or contain some form of entanglement. And more recently, Sulkowska group and Pavel Dabrowski, Tomasky, and Rystasiak analyzed the presence of knots in structures predicted by AlphaFold, and more entanglement was found by analyzing these structures. Now, first knotted protein was found. The presence of knots in proteins brought these three main challenges. On one hand, people had, mathematicians, biologists, and physicists had to start dealing with the problem of correctly identifying and classifying knots in proteinetic structures. And at the same time, the presence, the actual presence of entanglement in proteins brought this kind of question on how knots are created in protein during folding in the first place, since this seems kind of counterintuitive in some way. And more importantly, since creating knots in proteins seems to be a difficult task to achieve, why are there knots in proteins? Is there some benefit for the presence of knots in proteins? Is there some biological function coming with the presence of knots in proteins? Now, I think among these three big questions, the third one is arguably the most interesting one, and I'm really not going to talk about this at all. I just wanted to point to one of my favorite reviews on the topic. This was written by Sophie Jackson a few years ago, and the title is very self-explanatory: Why are there knots in proteins? And also doing a little bit of shameless self-advertisement. The subject here is also discussed at length in a recent review appeared last July in Physics Reports. What I'm going to talk about today has to do mostly with the first two questions. So, how do we identify knots in proteinetic structures, and also how can we understand a little bit better how knots form during the folding process. All right, so the first question, the first issue has to do with developing tools to characterize entanglement in open curves. As you probably know, classical knot theory deals with classifying the topological type of closed curves in space. And knot theories say that two knots to closed curves in space are equivalent if one curve can be continuously deformed into the other, whereby continuously I mean without allowing strand passages or any other topologically disruptive operation. Obviously, this setting doesn't work when dealing with open curves, because any open curve, complicated as you want, entangled as you want, can always be continuously deformed into the straight crossingless one. So traditionally, what mathematicians have been doing when dealing with entanglement in open structures has been artificially closing the open curve to form a closed one. And this operation can be done in different ways. One can decide to perform a single closure, perhaps by joining two ends of your spatial curve by the shortest line between them, shortest segment joining them, or one can adopt a more probabilistic or statistic approach by taking perhaps random closures defined in different ways. But regardless, the point is always to start from an open curve, closing it to obtain a mathematical knot, and then analyze the corresponding knot type. perhaps just once, or perhaps in a stochastic, in a probabilistic way. This has been the standard for many years. And the situation changed quite substantially a few years ago when Turaev introduced new topological objects called notoids. Now, in this setting, again, we can think about our favorite open curve in space, and we can decide to consider planar projections of this curve. And if we remember information about over and underpassing, we obtain diagrams of open-ended arcs, kind of in the same way as we obtain knot diagrams by taking projections of closed curves. And Turaev introduced this concept of notoids. There are just equivalence classes, defined as equivalence classes of planar projections of spatial open curves up to the classical Reidemeister moves and planar isotopies. And in this way, we were able to deal with open arc diagrams as their own well-defined topological objects. Now, these allowed people to study the entanglement type of open curves without having to rely on artificially closing the curve. And this can be done perhaps by looking at the S2 distribution of planar projections of your curve. So each direction of projection defined a different notoid diagram. We can analyze this notoid diagram using notoid invariant, defined in a similar way as our usual knot invariant. And if one consider all the possible S2 many projections, they end up with an S2 distribution of notoids that can be taken as a topological descriptor of the initial spatial curve. This point of view, this way of characterizing entanglement in open curves is one of the tools we're going to use today. All right, any questions so far? Are we all good? Great, okay. For the rest of the talk, I'm going to try and focus on possible approaches to tackle some of the questions arising when dealing with trying to understand the process of knot folding. And again, before going into the details of what we have done and going to the details of the results, I just wanted to give a further motivation to introduce the approach we decide to take. So the objects we're going to deal with are, again, spatial curve, open spatial curves, perhaps knotted or at least folded in geometrically complex way. Now, given a knotted curve or a knotted polymer, a natural question is to ask whether this polymer, this curve, will preferentially untie by a certain mechanism and why. And this question can be rephrased in a different way, and one could ask whether it makes sense at all to talk about optimal or minimal or preferred unknotting or knotting pathways for entangled curves. And having the topology lenses as a starting point, one can further rephrase this question as, does it make sense to formalize the concept of topologically optimal unknotting pathways? Is there such a thing as an optimal way to unknot an entangled curve from a topological point of view? Does it make sense to talk about optimal topological unknotting pathways or minimal topological unknotting pathways? And this is the first question that we tried and address. And a possible strategy to address this question and to find optimal unknotting pathways, topologically optimal unknotting pathways, is to define a measure of complexity for open curves and then look at what happens if one tries to follow the gradient descent induced dynamics under this measure of complexity. So in other words, we can define a way of measuring how complicated the entanglement of the initial curve is, and then we can look at those pathways that monotonically and optimally simplify this complexity, this measure of complexity. So we can look at a gradient descent dynamics based on this functional given by a measure of complexity. And in our case, this measure of complexity is going to be a measure of topological complexity. And this problem of defining a measure of complexity for open curves is something that Dimos and I started thinking about quite a few years ago when I was still doing my PhD. So this is this is started as this is an old project. It started a few years ago, and then Alex and I, together with Dimos, kind of resuscitated more recently. So at that time, Dimos and I were working a lot with notoids, and we decided to see whether we could use this notoid methodology to, again, find an effective way of measuring how complicated an open-ended knot, a knotted open curve was. And the starting point is the one I shared a few slides ahead. We can consider our spatial curve, knotted spatial curve, together with its S2 distribution of notoids. Now, an important feature, an important property of notoids is that, I'm sorry, it is possible to define a richer set of unknotting moves with respect to knots for notoids, and one of these moves are called forbidden moves, and they have to do with moving one endpoint of the diagram from inside the region to a neighboring one. And by looking at these forbidden moves, one can define the concept of a raveling number, so the minimal number of forbidden moves needed to fully untangle a notoid diagram, as suggested by the schematic here on the bottom of the figure. And this raveling number provides a measure of topological complexity for each single notoid type. Now, if we look at the S2 distribution of notoids for our initial open curve in space, and we analyze each planar projection in this S2 distribution, in each planar projection as a notoid type, and we compute their raveling number, then we can look at the average raveling number, or the integral over S2 of all the raveling numbers obtained as projections of the initial curve, and this gives us a measure of topological complexity for the initial curve. So a real number that tells us in some way how much effort is needed to create the given entanglement. And this is going to be the starting point to define our optimal topological pathways. So at this point, we would like to be able to look at the gradient descent induced dynamics defined by this average unraveling number, this measure of topological complexity. The problem is that we can't really differentiate, right? This is a very complicated quantity, a very complicated functional, and it's not like we have an analytical expression, and really we are not able to take derivatives here and actually follow a gradient descent. And this is where Alex and I implementing the next step, which was instead of doing what one would want to do, so take the derivatives and follow the gradient descent, we decided to explore the space of configuration, possible configuration of open knots using random Brownian force, and define these optimization procedure that allows us to move towards those configuration, minimizing the average unraveling number with the hope of approximating in unknotting pathways that was close to be the optimal from a topological point of view, from an unraveling number, average unraveling number perspective. And more precisely, our first attempt was to model our set of curves, our set of open curves as polymers using a semi-flexible Kremer-Grest polymer model inspired by DNA modeling. And then starting from each model configuration, modeled knotted configuration, we would compute the average unraveling number, and then we would apply random Brownian force to each bead to perturb the polymer. We would do that with a number of independent runs with different random seeds to have like an idea of the landscape of the possible neighboring configurations of our initial curve. We would compute the average unraveling number for all of these configurations, and then we would move towards the configuration, minimizing the average unraveling number. And then we would keep repeating this procedure until hopefully achieving total unknotting. So again, the idea is we start from our initial knotted configuration. Using this physical model, we explore the space of neighboring configurations, and we move towards those configurations that minimize the average unraveling number. And we keep doing that until hopefully we get to something that's totally unknotted. This is an example of a pathway that we found starting from a deeply knotted open-ended trefoil. As you can see, or at least as I hope you agree, it looks quite neat. The unknotting seems very fast, and there doesn't seem to be any unnecessary movement. All the movement seems to be optimized towards undoing the knot, towards undoing the entanglement. You can see like the two ends, they consistently move towards the knot core towards the knot part, and they kind of undo the knot at a constant speed. Now, this one can be quantified by looking at different runs of these numerical experiments, numerical simulations. And as you can see from this picture, if we compare these approximated optimal pathways with the result of average physical simulation, which often result in unknotting, we can see that unknotting by these monotonic, these optimal pathways faster than the average physical simulation. And also, as you can see from the green curves showing the behavior of the average unraveling number against the time steps of the simulation, you can see that these pathways are kind of robust. So even if we are approximating these optimal pathways, and obviously each pathway is going to be unique, we can see that we can be reasonably happy with the fact that we are really getting close to a pathway that is the optimal one, since all the runs are quite consistent with each other, and these pathways seem to be quite robust. And moreover, we do achieve what we want. We have these kind of pathways with a always decreasing, monotonic decreasing topological complexity. So there's no need to make the configuration more knotted in order to untangle it. We can move towards total unknotting in a monotonic way. Now, at this point, since we had these technology, these tools that allowed us to kind of steer the simulation topologically, to push the simulation, to drive the simulation with topological quantities, a natural next step was to, what about the to ask ourselves, what about the other way around? Can we use the same functional, the inverse of the same functional, to create entanglement rather than undo it? So we tried to explore a little bit this question using the same model as before. And we ran a few experiments, and we were initially quite surprised when noting that knotting was pretty consistent. So in this experiment, we would start from unknotted configurations, let them move randomly until something with non-trivial average anomaly number was found. And then since that, so as soon as a configuration with non-zero average anomaly number was found, then the functional push would start to kick in, and we would start to move towards those configurations, maximizing this topological complexity rather than minimizing. And what we found was that basically the first thing that would happen in this simulation was a single twisted loop was formed, and then the chain would start to the one end of the chain would start to pierce this twisted loop that just formed, and then it would keep piercing it repeatedly, recursively, forming alternating torus knots of increasing complexity. So first the trefoil knot, then the 5-1, then the 7-1, and so on. And again, this behavior was very, very consistent. It would happen all the time in exactly the same way. At the beginning, the first non-trivial configuration would be like a twisted loop, and then we would have these increasingly complex repeated piercing of the loop, creating non-alternating torus knots of increasing complexity. And we weren't necessarily expecting this to happen, so we started asking ourselves why. And we think that likely the reason is that has to do with mainly how the function is defined. Again, is defined by looking at these unraveling numbers, right? The average unraveling number, looking at this complexity defined by forbidden moves on autoids. So this repeated piercing of an already knotted configuration is the fastest way to increase the average unraveling number, the global topological complexity, the global knot complexity under this definition. But also, this repeated behavior was really possible because the initial configurations were of a certain kind. Initially, we would have these very simple twisted loops that would, promote, would push for this continuous piercing behavior and the formation of these, again, increasingly complex alternating torus knots. So said in a more brief way, we think that one of the reasons why we were observing this behavior had to do with the local geometry, local geometric properties of the configurations achieved, allowed by the physical modeling, achieved by the physical modeling at the beginning of the simulation, and then obviously also by the definition of our topological complexity. Now, if you like knots in proteins, as I do, you know that alternating torus knots are really not a thing for proteins if we forget about the trefoil knot, because all the knots in proteins, at least all the knots in experimentally determined protein structures, are twist. So again, we do have one alternating torus knot, which is the trefoil, which also happens to be a twist knot. If you don't know what twist knots are, here is a figure, is a schematic representing them, and they're just those knots created by taking an unknotted loop, twisting it a number of times, and then clasping the two ends of the loop twist together, as shown here in the picture. And again, it's a fact that all the knots in proteins, all the known knots in proteins are twist knots. And people have been thinking quite a lot why and how, why do we only have twist knots, and how do these twist knots form in the first place in proteins. And in the literature, you can find different approaches to this question, and some of them are very mathematical in nature. One of the first explanations was due to Taylor, the same person that found the first deeply knotted protein structure. Taylor conjectured this hairpin theory, in which basically at the beginning of the folding process, a twisted loop is formed, and then there is a single piercing of this twisted loop by one of the two ends of the proteins, and in this way, you can only create basically twisted knots. So these mathematically inspired folding theory explain the absence of non-twist knots. Now, these work quite well at the beginning, and then, as it usually happens with real world situation, new experiments came by, new simulations came by, and the variety of configurations you can find quite increased over the years. So after a few years, this theory stopped agreeing with simulations and experiments, and new and more rich theories were proposed in the literature. In particular, I want to mention one of my favorite ones, due by Flapan and collaborators in 2018, where they provide this topological description of knot folding inspired by experimental evidence. I don't want to go into the details of this theory, but it's a beautiful paper, very easy to read, and it's quite intriguing and convincing, I think, what they are proposing. And again, this topological inspired description of knot folding explains, again, the absence of non-twist knots. The approach we decided to take on this question was grounded in this idea of looking at optimal folding and unfolding of knotted polymers, and try to see how the constraints of the system would change the landscape of knot populations you might achieve. And our initial guess, our initial conjecture, our driving idea was that perhaps geometric constraints of different proteins really matter and do play a key role in shaping the landscape of possible knots formed by proteins. And this is what we tried to verify and check. And okay, so our guess was that geometric constraints of polymers, of proteins in particular, really matter. And the main reason why we thought that is that proteins specifically are really not they're really not random polymers, right? They're really not random curves. On the contrary, they have quite a constrained local geometry, and the constraints of this local geometry can be quantified in terms of the local bending and dihedral angle phi and theta. I don't want to go into the definition of these angles. A schematic of what they are and how they can be defined is shown here on the top left of the slide. The point is that the local geometry of proteins is quite constrained, and the distribution of possible bending and the dihedral angle in protein structure is highly biased, highly skewed, and this in turn results in having highly skewed curvature and torsion distributions, as shown here by the figure. So they're really not random. They have quite constrained local geometry, which in turn, results in quite constrained overall 3D geometry. And our guess was that these constraints bring important biases on the topological side. So bring important constrictions, bring important obstructions to the creation of certain knot types. And to test this hypothesis, we decided to change a little bit model to look at our optimal topological pathways, since, you know, including geometric constraints in our first initial polymer model seemed a little bit complicated at the time, and a simpler approach to this task was to use a growing self-avoiding walk. In this model, again, our polymer is made up by separate beads, and we grow polymers by placing the new sphere according to the two angles phi and theta. And if we start from a short configuration and we start growing it, again, if we want to explore a little bit the possible set of configurations this curve might take while growing, we can grow it a little bit by applying a different by applying a number of independent runs in which each new bead is positioned in a random way by sampling phi and theta from given distributions of angles. We can compute the corresponding average unraveling number, and we can move towards those configurations, maximizing the average unraveling number. So just to repeat, we can grow our polymer by starting from a short configuration, exploring the possible ways in which this short chain can grow, and then pick those configurations going towards maximal average unraveling number. And each time the independent runs, the next neighboring configurations we're exploring are picked by picking these pairs of angles, phi and theta, every time we add a new bit, every time we add a new sphere. Okay, now,
question.
yes.
The distribution
ahead.
for the random angles phi and theta, are those distributions taken from data, from experiments, or from real proteins?
Yes, I'll tell you in a second. So we can do different things, right? We can choose phi and theta completely randomly from uniform distribution, and in this way we can get a self-avoiding random walk. Or we can try to create a model similar to the one we had before. So we can take phi according to some Gaussian distribution about pi to add some sort of persistent length to have something that's polymer-like. Or we can choose phi and theta according to a protein-like distribution to get a protein-like polymer. And in this case, what we did specifically was look at a number of non-redundant protein structures, get the distribution of these angles, and then we would pick these distribution of angles are paired. We would pick randomly the two angles, the pair of angles from these paired distributions. Does it make sense? Does it answer your question?
Yes.
Perfect. Okay. And here I can show you a couple of examples. So if we choose this protein-like model, this is like what a short configuration of 100 bits can look like. And as you can see, obviously it's not like it looks like a protein, but there is some protein-like feature we can observe. And on the other hand, this is like a polymer-like configuration where phi and theta are chosen, where pi is chosen from a Gaussian distribution about pi to give this kind of persistent length. So you can see the significant differences in what you get if you change the way in which you are choosing the angles. And then our next step was to check, how these different choices affect the kind of knots you can get, using our optimal knotting pathways. And we were very happy to see that indeed, by adding these protein-like constraints in different ways. So let me go back, sorry, to this figure. We can choose these angles, like taken from the protein distributions in a fully random way. So we have the two distributions coming from experimental data from experimentally solve protein structures, and we get something that looks like that. Or we can also decide to go back to the 2 peaks of the distributions. Here you can see these 2 peaks in the bending and twist angles, and the 2 peaks correspond to alpha helices kind of angles and beta sheets kind of angles. So we can decide to take angles only from helices, or maybe exclusively from non-helical portions of chains. So to make something that's kind of like a protein, but without helices, or which is kind of like a protein, only contain helices. Obviously, this is not very realistic, but it was a good way of testing the different possible biases we might get. And getting back to the result picture, we can see that from these figures here, we can see that after we add these geometric constraints, we see a substantial increase in the formation of twist knots. If you look at this figure, we see that, we observe again so each of these plots correspond to a different choice of angles. The leftmost plot is the unbiased, what we call unbiased growing model, in which our curves are protein-like. So phi is taken to give this persistent length, and theta is chosen randomly, uniformly. The mid plot is where we are picking angles in a protein-like way, and the rightmost plot is where we are picking angles in a protein-like way, but only from the helical side of the distributions. And in all of these 3 models, we see that we repeat these, we see again this behavior of creating initially trefoil knots, the light blue band, and then the Storz knots as the chain keep growing, become often increasingly complex alternating torus knots. So the first become 5-1, then 7-1, and this is described by these bands of the trefoil slowly becoming 5-1 and then 7-1 as the chain becomes longer. But importantly, we see this this increase in the occurrence probability of twist knots, and also of complex knots that are not alternating torus knots, or connected sums of alternating torus knots. And the difference might seem small, but is actually statistically significant, as we can see from these 2 other plots. And it is quite substantial. Again, the preferred behavior promoted by our functional is this behavior of creating increasingly complex alternating torus knots. But if you constrain the geometry in a protein-like way, then configuration that allow for the formation of twist knots become more likely, and you start to actually see them. And the same behavior, can be also observed if you forget about the topological steering, if you just grow these models in a random way only by picking different angle distributions. But the probability that such a chain, a growth chain, a chain becomes knot is much, much lower. So it's much, much, much more difficult to observe and show that this increase in the presence of twist knots is statistically significant. So using this topological steering, using this knot functional, allows us to increase by a lot the probability that our curves are knotted, obviously, and we amplify these differences induced by constraining the geometry of the polymer we're growing. And this is our main result. We are showing that these geometric constraints indeed promote the formation of twist knots. And just to conclude, I'm also 45 minutes, so almost reasonably on time. What we think we are bringing you with this project is that we are showing that it does make sense to talk about optimal topologically optimal unknotting or knotting pathways. And we can do this effectively by looking at different ways of defining knot or topological complexity. And this, idea of, looking at topological complexity as functionals can be quite effective in studying the folding and unfolding of knotted polymers. And also we think that, our last results, they provide quite compelling evidence that the geometric constraints are one of the key factors in shaping the knotting spectrum observed in nature. Now, obviously, the kind of knot spectrum we are finding is far away from what we find in nature. There's still this high prevalence of alternating torus knots. And this has to do with a very simple mathematical formalism that can definitely be improved. One might strive to find the actual, optimal pathway by finding ways of computing gradient descent, so defining some sort of derivative, some sort of directional derivative for our complicated functional, and then, move towards the actual, if it exists, the actual unique optimal minimizing pathway rather than doing this probabilistic search. And also, we're not using physically realistic models, right? So by improving these two aspects, I think one could get closer to find the actual, to replicate the actual knot population found in nature, and explain one of the factors responsible for how, why, and which knots in proteins are formed. I'm just going to finish now. I want to acknowledge again the contributions of Alex and Dimos. And again, I really care about this project because, it's something that started so long ago, so many years ago, with Dimos, and the way in which Alex and I and Dimos resuscitated it a few years ago, I think was, quite nice. It was fun working with them, and I think we're all happy about the results. So I'll just thank you for the attention, and I'll stop here. Do you mind if I stop sharing for a second so I can see people? I'm happy to share again if needed.
Okay, Agnes, thank you very much for a very interesting talk. Floor is open for questions. I've got a question. Could you put your main result on, the one that shows sort of knot probability
Yeah.
knotting, twist knots versus knotting, given that it's knotted, the fact there would be twist. And then the next one.
Yeah.
Yeah, that one. So the knot probabilities are going up toward, the twist probability is going toward, what, 10%, given that it's 80% knotted.
Yeah, that's correct. So again, the proportion of non-alternating torus knots is quite small, but it's start to be, small but quite substantial once you add these constraints. We also see, I haven't touched on that, but we also see that after adding these constraints, the probability that the configuration gets knotted increases a little bit. We think this has to do with the fact that by adding these constraints, the configurations become spatially more confined. And so this is like a byproduct of them being more confined. But even if you factor by this thing as shown on the rightmost plot, still the number of twist knots is significantly higher after adding these constraints.
You were also mentioning confinement. So you're forcing confinement by the choice of these angles somehow?
Yes, we're not, we are measuring it a posteriori. So we just checked whether, adding these constraints was like increasing confinement. And it is a little bit. I mean, not greatly, but it is a little bit. And we think that this might be one of the factors explaining the increase in knot probability. But it is done a posteriori, just to try and explain this increase in knot probability. kind of makes sense, right? If you have something more confined, if you are forcing the geometry to be a little bit protein-like, right? You're forcing the geometry to be more constrained rather than, these persistent pathways or random pathways. And it does make sense that your chains tend to stay in a more spatially constrained.
It should also promote knotting itself.
Exactly, that's what it does. Yes, exactly. But even if you factor for that, so even if you take into account the difference in knot probability, still twist knots are more likely than.
Yeah. Hi, I've got a couple of questions, if I may.
Yes.
Thanks. Thanks for the super interesting presentation. Really loved it. So I guess the first question is, do you know of another knot invariant that could bound the AUN from above? Because, yeah, clearly it's a very complicated integral over all projections, so that's going to be very hard to understand how to do gradient descent. But would there be another way of just bounding it from above that you know?
So to be honest, I haven't thought about that. So without thinking too much about making it more simple, you can achieve a forbidden move. Let me go to, you can achieve any forbidden move by 2 crossing changes, right? So I haven't thought about it. So yes. So not answer your question, but to go towards like a simpler, definition of this topological complexity of not complexity. Years ago, when Dimos and I started, we also looked at functional defined by the inverse of the radius of gyration and by the inverse of the average crossing number, I think. And maybe we also looked at different like geometric quantities. And Dimos, correct me if I'm wrong, but I kind of remember that the inverse of the radius of gyration was doing something quite interesting and was able most of the times to unknot configuration in kind of a weird way that obviously very, very fast to compute. It's not necessarily easier to differentiate, but faster to compute. While the average crossing number failed quite consistently. And this kind of makes sense if you think about the presence of hard knot diagrams, right? So knot diagrams that cannot be simplified before making them more complicated from a crossing number point of view. So if you try to unknot by monotonically decreasing the average crossing number, it shouldn't be too unlikely to get stuck on local minimal configuration and then you can't finish unknotting. But it was years ago and we haven't like resuscitated that part of the project. So I'm not sure if I'm telling you the truth or not, or if it's just false memory.
Yeah, no, clearly obviously it's a hard subject, but yeah, it'd be interesting to try to get a simpler way of getting some gradient descent to be doable.
Yeah.
So bounding it from above by something differentiable might be a way.
Yeah, it could be nice. Something else that I would like to work on, and again, it's not necessarily more simple, and I think your idea is probably more effective than what I thought, but there is this there are these newish, not really new theories stemming from seminal work by Sturm on geometric measure spaces, and they allow to define like derivatives on structure objects, extending the concept of networks and higher order networks in a geometric measure theory setting. And if you have functionals on these objects, then you can define derivatives. And I think one could possibly tweak a little bit this theory to include spatial curves to see spatial curves as some sort of, geometric measure spaces in such a way that you can maintain the constraint given by ambient isotopes, and perhaps that's a way of going towards like a formal definition of differentials of topological functionals. But
yeah,
I'm not sure how much is doable.
Yeah, and that leads me to the other question that I had, which is, could you think of maybe a curvature flow that could lead to unknotting? And like that, so that's kind of similar to what you were saying, but like, because I was thinking with the phi and theta, you could try to like wiggle the phi so that it tends to pi, and then the theta tends to 0. Like
possibly.
That's kind of, I mean, you could do that in like a discrete curvature flow type setting, and that would be, I mean, in that setting, you could use something like metric measure space.
Yeah, I'd love to talk more about this. It seems plausible. It seems like reasonable. And yes, I think, there are definitely ways of making this much better and more formal. And to be honest, what you suggested seems very intriguing. So I'd be happy to chat more about this.
Oh, and just a final one, if I may. Could you share that reference from AlphaFold not being able to distinguish topology again? Because that went by a little bit.
Yes.
Thanks. Just, I guess, AlphaFold blindness, and I can find it.
Yes. This is by Pavel and Andrei. Can't remember, it must have been like 1 or 2 years ago. And the other paper is by Surkovskaya group, and they created this replica of the knot-plot database called AlphaKnot, in which they analyze AlphaFold predictions instead of PDB structures, and they do the same thing, right? They take the knotted ones and they classify the entanglement, I think in the same way as they did in knot-plot, but at a much, much, much larger scale.
Okay, excellent. Thank you so much. It was a super interesting talk. Yeah, I'd like to, if
Thank
you're
you.
interested, we could keep in touch about this ideas later. So.
Oh, definitely. It's not the best time ever for me to keep in touch with people because I'm due to give birth in 7 weeks. But yes, I'd love to eventually.
In the future,
yes.
Time. That's fine.
Yes. In the next few weeks or in a few months.
Hello, Frederick speaking. Can I ask a question?
Yeah.
Yes. Yeah. So thank you for this very interesting talk. So I was wondering, so did people try to understand what it means to knot a protein in terms of energy? Because of course, when you do the knot, you may favor some contacts between the chains. On the other hand, this is clearly a hindrance for dynamics. And so it has to contribute to an entropic penalty. And so I was wondering whether some studies have addressed this balance between possibly favorable contacts on the one hand and possibly an entropic penalty on the other hand, to understand the metastable states of the corresponding to the knots which are seen in the protein data bank.
So I'm not 100% sure if people have done exactly what you're saying. My guess would be probably at least partially yes, but to be honest, I'm not 100% sure. Again, doing a shameless self-promoting, I think if there is an answer to this question, you can find it in this review here. Where is it? Topology in soft and biological matter. It's quite recent. It came out on last July. And all the topology in soft and biological matter, whatever, is extensively discussed, and this includes folding and functioning and everything on knotted proteins. There are a couple of chapters dedicated to this stuff, and we have, obviously it's a review, so we have tons of references, and I think the answer of your question could be found here. I'm sorry, I don't have any more precise answer than this.
Yeah, so indeed, so while listening to you, so I was spotting entropy in the survey, and so figure 50 indeed is possibly of interest. So I'll have to take a more quick. Okay, thank you for the pointer.
Perfect.
Yeah, very interesting. Yeah, thank you.
Thank you.
Okay, if there are no more questions, let's thank Agnese again for a great talk.
Thank you. Oh, and also, yes, Alex is speaking about the same stuff in a week at APS meeting as written in the chat. So you can have a do-over. And Alex's slides are much nicer than mine.